Okay, hydrolysis of starch. Starch can be hydrolyzed by strong acids, high temperatures, or enzymes by combination of both. Dextrins comes first, which are gradually broken down into oligosaccharides, maltose, and finally glucose. Can't trehalose form. When we do hydrolysis of starch, what is the basic building block of starch? What is the basic, I mean the primary building block of starch? Is it glucose, fructose, or lactose? What is that?
Glucose.
Yes, glucose. Then the final product of the hydrolysis of starch will be glucose. Okay, then I don't know whether trehalose can be formed from glucose. There are some mechanisms, but upon the hydrolysis of starch, we cannot assure that. Okay, it will be broken down into its monomers. I mean the basic building blocks. And after that, if there's conditions fair desirable for the reactions, trehalose or other two carbon or 3 carbon products can be formed, but in the hydrolysis of starch we can assure that. Okay. Okay. Then yes, in this experiment you have to take 0.5 grams of starch into a mortar, and you have to add a few milliliters of cold water and mix with the pestle. You know the mortar and pestle. So you have to pour this mixture into a small beaker with half full of boiling water. After that, you have to add 2 to 3 milliliters of concentrated acid. In here, we use acid hydrolysis, and we are going to use concentrated acid for that. Okay, immerse the beaker in a boiling water bath, and we provide high temperature and maintain boiling until end of the experiment. Okay. Note that the time of the heating commence, and at intervals there's small portions with iodine solution and with Fehling solution. Note that the liquid should be neutralized for Fehling test. Okay. Then we can see, or we can observe the hydrolysis process. In the hydrolysis of starch, it will within a short period of time it will not produce glucose, and it will break down into dextrins and then maltose and finally to glucose. Then that will take some time. Okay, so when we do when the hydrolysis is proceeding, we can, using the iodine test and Fehling test, we can observe the changes, I mean the products. Okay, you know that starch will give blue black complex with iodine, but black blue complex cannot give by maltose as well as glucose. Okay, then you know that starch is not a reducing sugar, but maltose and glucose are reducing sugars. So if we test that for that use in Fehling test, starch will not give positive results for Fehling test, but on the other hand, maltose and glucose will give. Okay, so in this experiment we test that property. I think you have done that in your science classes. We hydrolyze starch and take small drops into a porcelain tube, and we test for iodine and Fehlings. Okay, so same thing we are going to do here. Okay, then what if you compare acid hydrolysis against enzymatic hydrolysis, which will give which is faster, or which is more effective, or which will give glucose within a short period of time, or which is faster than if we compare these two. What is your opinion? Is it enzymatic hydrolysis or acid hydrolysis?
Enzyme?
Yes, enzymes are, you know that you have learned that in biochemistry, enzymes are more specific, and they have designed naturally in order to do specific biochemical reactions. So enzymatic hydrolysis is more effective than acid hydrolysis. Okay. Okay, then I will show you the video. This is very basic experiment, and if you have any questions after that, you can raise.
Today our practical is hydrolysis of starch, and for that we have already measured 0.5 grams of starch, and first we are going to add it into a mortar, and I'm going to add few amount of distilled water. I'm going to dissolve it properly. And then I'm going to add this mixture into a boiling tube half filled with hot water. And then I'm going to add 2 milliliters of conc HCl. And then I'm going to place this boiling tube in a water bath. And we are going to maintain this water bath until the end of our experiment. You have to note down the time which we put this boiling tube into the water bath. Now we are going to test few drops from solution which is in the water bath. Then we are going to take a drop and add iodine, and likewise we are going to add Fehlings into another drop. Then observe the color change. Then wait for 2 minutes, and after 2 minutes repeat the same procedure. Likewise we are going to test these drops with iodine and Fehlings, and we are going to observe the color change of Fehlings and iodine.
Okay, here are some products of during the hydrolysis, starch hydrolysis products formed during the starch hydrolysis process. Starch, amylodextrin, erythrodextrins, and acrodextrins, and these are uh, dextrin types. Okay, and these dextrin types has different properties. Okay, they are unique properties. For an example, amylodextrin uh, is will give purple color, but erythrodextrin will give red color for iodine test. Okay, but acrodextrin will not give any color to iodine test. Okay, so there will be a series of product as I mentioned earlier in this starch hydrolysis, and the final product will be glucose. Okay, and what can you say about the reducing properties of these dextrin products? Are they non-reducing agents or reducing agents? What can you say about that? Will they act as reducing agents? What is your opinion? You know, starch is not a reducing agent. What can you say about the dextrins? Dextrins, all the dextrins contain some carbonyl groups. Then technically they can act as reducing agents, but not that strong as maltose or glucose. Okay, technically they can give positive results for Fehling test. Okay, all the dextrins contain some form of carbonyl groups. Okay, then when you, I don't know whether you can remember, check if I am telling you lies. Your science syllabus in ordinary level, we have to do the starch hydrolysis using enzymes in saliva. Okay, there we add sodium chloride and citrate. Okay, in there we just put our saliva to starch, and then we add sodium chloride and citrate. What is the reason for adding sodium chloride and citrate? We have learned these things. Okay, even I have done your syllabus. We are the first to start advanced level and ordinary level syllabus. Then after that, I think we have done the same syllabus. Okay, then why we add sodium chloride and citrate when we doing enzymatic hydrolysis? In our syllabus, biochemistry, for your course, we don't have to cover enzymatic hydrolysis. Okay, but if there's it's good. Okay, then why we add sodium chloride and citrate? Chloride can act as an activator for amylase enzyme, as well as this reaction is well occurred in the pH range of 5.6 to 6.9. Okay, close to neutral pH. So sodium chloride and citrate can act as a buffer. Okay, so that's why we add the sodium chloride and citrate or citric acid, which is a weak acid, when we doing this enzymatic hydrolysis for starch. Okay, so that's it. Then I will move on to the next experiment that we need to cover. We have discussed a series of qualitative analysis for qualitative experiments for carbohydrates. Then I will move on to quantitative analysis of carbohydrates. Okay, in here, you know the difference between qualitative and quantitative analysis. In here, we are going to analyze carbohydrates quantitatively, specifically glucose. Okay, then the principle of this test we have already discussed, that is in the Fehling test. And this method or this titration is known as Lane-Eynon method. Lane-Eynon method. pH range of previous experiment. Then when we do the enzymatic hydrolysis, the pH, the favorable pH is about 5.6 to 6.9. Okay. In here, in the previous experiment, we don't need to provide that pH. Actually, that pH cannot be there because we did acid hydrolysis. Okay. I just mentioned about the enzymatic hydrolysis. Okay. Nilesha Jarap, then what's your question?
Sir, previous one के sir sodium chloride वे citrate එකතු වෙලා මේ combine වෙලා buffer එක හදනවා කිව්වේ.
ඔව් ඒක මම ඇත්තටම කිව්වේ ඔයගොල්ලෝ බලන්න ඔයගොල්ලන්ගේ මේ O level science පොතේ සමහරලාට මට මතක විදිහට තියෙනවා. අපි එතනදී මේ acid hydrolysis වලට අමතරව enzymatic hydrolysis test කරනවා. එතනදී අපි කෙල දාලා වෙලා starch වලට අපි sodium chloride උයි citric acid උයි දානවා. ඒ sodium chloride citric acid උයි දාන්නේ ඇත්තටම එකක් තමයි chloride ඒක ඔයගොල්ලෝ මතක ඉගෙන තියෙනවා මේ digestion of carbohydrates වලදී chloride amylase activate එකක් විදිහට use වෙනවා. අනිත් එක තමයි මේ citric සහ sodium chloride වලදී මේ more actually it's not similar to buffer action. It can provide the favorable pH for this enzymatic reaction. Okay, you know that in every biochemical reaction there's a favorable pH range. Okay, so the favorable pH for this acid enzymatic hydrolysis of starch is 5.6 to 6.9. So it is closer to neutral pH. Okay, so when we mix sodium chloride and citric acid, we can have that pH. So that's why we use that. Okay, so that's what I wanted to tell you. It's out of this.
Sir,
Okay. Yes.
so actually my question is that sodium chloride act as a conjugated base?
Sodium chloride act as a conjugated base. To some extent, yes, but actually, what is the conjugated base of citric acid? What can be the conjugated acid for citric acid? අපි බෆර් එකක් කියලා කියන්නේ සාමාන්‍යයෙන් weak acid එකකුයි එකේ conjugated base එකයි නේද? දැන් අපි කලින් කතා කරාද buffers ගැන මෙතන නේ? එතකොට මේ citric acid කියන එකේ conjugated base එක විදිහට ගන්නත් පුළුවන්. එහෙම කිව්වත් වැරදි නැහැ මෙතන. එහෙම වෙන්නත් පුළුවන්. අපි මේ දෙක එකතු කරාට පස්සේ මෙතන ඉන්නේ citrate buffer එකක් form වෙන්නේ. ඒක අර pH එකට ගොඩක් දුරට සමානයි. හරිද? නිලේෂා.
Okay, sir, thank you.
Okay, then if you have any questions, please ask. Then if I can help, I will definitely support you. Okay, then quantitative analysis of carbohydrates. So this method is known as Lane-Eynon method. Okay, in here we are going to analyze glucose concentration of a given food sample. Okay, we have to adhere to some assumptions here, but assumptions when you use Lane-Eynon method, but this is widely used in chemical experiments. Okay, so this if we do not have advanced test kits to analyze glucose, then we can use this Lane-Eynon titration as a preliminary experiment to analyze glucose percentage or concentration in a given sample. Okay, this is a quantitative test which is used to estimate the glucose content in a given sample by doing a titration. The titration is based on the oxidation and reduction capacity of methylene blue indicator and glucose and glucose. Okay, you know that glucose is a reducing sugar. It means it can act as a reducing agent, and glucose can be further oxidized while the oxidizing agent will be reduced. Okay, then in this titration, we use Fehling's A, Fehling's B solution, methylene blue as the indicator for this test. And when we use methylene blue as the indicator, you should know that the property we use as the indicator is different. Okay, and the sugar solution or the food sample that we have provided with or to be tested. Okay, I'll go through the experiment, and please understand this experiment very clearly. This is only quantitative analysis we discuss in this under carbohydrates, and usually this can appear in your exam. Okay, so I just want to tell that also. So you have to take the prepared standard sugar solution or the sample we are provided with, and pipette 5 milliliters of Fehling solution A and 5 milliliters of Fehling solution B into a 100 millimeter conical flask, and add 2 to 3 fumarate stones. And you have to gently boil the content of the flask for 2 minutes, and add 1 milliliter of methylene blue indicated solution. Run the sugar solution from the burette without interrupting boiling. Hold the burette in hand above the flask and add sugar solution dropwise. Do not keep the burette above the flask continuously as it may get heated. Okay. In this titration, we have to take the sugar solution into the burette. Okay. So that's quite different than the usual titrations, because in usual titrations we take what is to be tested in the conical flask, and usually we add acid or base using the burette. But in here, you have to take the sugar solution or the sample to be tested into the burette. Okay. Add the sugar solution until the content in the conical flask becomes colorless. Note the endpoint, and this will be an approximate endpoint for your estimation. Pipette 5 minutes of Fehling's A, and then the first titration is done, completed. And after that, you have to again pipette out 5 minutes of Fehling's A and 5 minutes of Fehling's B solution into another 100 milliliters conical flask, and you have to add 2 to 3 fumarate stones into it, as we done previously. Gently boil the content. While boiling, run the sugar solution to 3 to 4 milliliter less than your approximate endpoint. After that, you have to add 1 milliliter of methylene blue indicator solution, taking care not to allow touching the sides of the flask. While the contents of the flask begin to boil, add sugar solution again from the burette until the blue color of the indicator just disappears. The titration should be completed within few minutes. Okay. Then the difference between the first titration and the second titration is that in the first titration we do to get an approximate endpoint. Okay. In the second titration and the other subsequent titrations, we know that the approximate endpoint. Then 2 to 4 milliliters before the approximate endpoint, then only we add methylene blue indicator. Okay. First titration එකන්නේ approximate endpoint එකක් හොයාගන්න. approximate endpoint එකක් හොයාගත්තට පස්සේ දෙවෙනි තුන්වෙනි සහ හතරවෙනි titrations අපි endpoint එකට 2 to 3 milliliters පල්ලෙහා වෙනකල් sugar solutions add කළා. ඊට පස්සේ තමයි අපි indicator එක දාන්නේ. Okay. There is reason for each and every step here. Okay. In here, we use this assumption that 5 milliliters of Fehling's solution A equal to 50 milligram of glucose. Okay. This is a kind of conversion factor. Okay. When we doing the calculation, this is required. 5 milliliters of Fehling solution A is equal to 50 milligram of glucose. Okay, then I will show you the video, then we can discuss further. Please watch this video carefully. As I mentioned earlier, this can appear in your exams. Okay.
Today's practical is glucose estimation. For this practical, we need Fehling A and Fehling B and solution and methylene blue indicator and fumic stones. First of all, we have to pour the sugar solution into the burette. Then we need pipette out Fehling A solution. Next, we should pipette out 5 milliliters of Fehling B solution into the conical flask. Then it should be gently boiled for 2 minutes. Create a fumic stone for this solution. Then add 1 milliliters of methylene blue solution. Then add sugar solution into the this solution until this solution become colorless. Here you may see the bubbles become colorless. Then you have to take the readings and it will be the approximate endpoint. Then we have to repeat previous procedure to get another reading. Then you have to add sugar solution into this solution. Then add methylene blue into the solution. Then we have to repeat the step to get another reading. Then add 1 milliliters of methylene blue. Add the sugar solution until the bubbles become colorless. You may see the bubbles become colored.
Okay, can you guess what is happening here? What is the redox reaction, main redox reaction happening here? සරල දෙයක් තියෙන්නේ අපි ෆේලින්ස් A වලින් මිලිලීටර් 5ක් හයිටම් යනගෙන සහ ෆේලින්ස් B වලින් මිලිලීටර් 5ක් කොනිකල් ෆ්ලාස්ක් එකකට දාලා පොඩ රත් කරලා ෆියුමස් ස්ටෝන්ස් දාලා ඊට පස්සේ අපි බියුරේට් එකේ තියෙන සුගර් සොලියුෂන් එක ඇඩ් කරනවා. මෙතනදී මෙතිලීන් බ්ලූ එක මෙතිලීන් බ්ලූ ඉන්ඩිකේටර් එකක් විදිහට පාවිච්චි කරනවා. එතකොට මෙතන වෙන redox reaction එක මොකද්ද? What is the redox reaction happening here? There's a reducing agent as well as a oxidizing agent. Then what is the reducing agent here?
Glucose.
Yes, glucose. Reducing sugar. In here, we assume that the sample contain only one type of reducing sugar, that is glucose. Okay. So usually we do this titration for glucose-rich samples. Then we can assume that the only reducing sugar type is glucose. Okay. Then what is the oxidizing agent here? The main oxidizing agent?
Fehling solution.
Is Fehling solution. Then what is the active component of Fehling solution? Specifically CO2 plus. Yes, yes, CO2 plus. Okay. Then you know the redox reaction here, the main redox reaction. CO2 plus will reduce into Cu plus, and the sugar solution, glucose, will oxidize into gluconic acid. Okay. Then how can we determine an endpoint? Then how can we determine an endpoint? And also we use a conversion factor here. We think that 5 milliliters of Fehling's solution A is equal to 50 milligram of glucose. Okay, in here, please understand that carefully. We to the conical flask we add only 5 milliliters of Fehling's A. Okay, even in the practical video, we have added Fehling's A solution using a graduated pipette, but it's better to use a bulk pipette there. Okay. In order to increase the accuracy of this test, we have to measure exactly 5 milliliters of Fehling solution. It's okay to use measuring cylinder or graduated pipette to measure Fehling's B, but we have to be very serious on measuring Fehling's A, because of the accuracy of this experiment for the calculation depend on that. Okay, we use this conversion factor. 5 milliliters of Fehling solution A is equal to 50 milligram glucose. Okay, then we add sugar, I mean the glucose, to the conical flask until all these Fehling solution A means Cu2 plus consumed. Okay, after all the Cu2 plus that is in the Fehling's A consumed, methylene blue will reduce into leucomethylene blue. Okay, methylene blue is, you know, what is the color of methylene blue? What is the color of methylene blue indicator? Is it red? Yes, blue. Blue color. Methylene blue is blue color. Then at the end point, methylene blue will convert into leucomethylene blue. It's a kind of reduction. Okay, methylene blue will reduce into leucomethylene blue. Methylene blue is in blue color and leucomethylene blue is colorless. So that is the end point. Okay, සරලව කිව්වොත් එහෙම අපි මෙතනදී හරියටම Fehling's A පහක් මැන ගන්න ඕනේ. ඒක අපි දානවා conical flask එකකට. එතකොට අපි උඩින් add කරනවා reducing sugar, you can glucose. එතකොට Fehling's A වල තියෙන Cu2+ Cu+ බවට පත් වෙනවා. එතකොට මේ Fehlings මිලිලීටර් 5ෙම තියෙන Fehlings Cu2+ ඉවර වුණාට පස්සේ ඉන්ඩිකේටර් එකේ තියෙන methylene blue dichomethylene blue වෙනවා. ඒක තමයි endpoint එක. ඒ කියන්නේ මේ මිලිලීටර් 5ෙම තියෙන Fehlings මිලිලීටර් 5 Fehlings A වල තියෙන Cu2+ ඔක්කොම consume වුණාට පස්සේ consume වෙන නැත්නම් consume කරන්න නැත්නම් reduce කරන්න අවශ්‍ය වන sugar ප්‍රමාණය තමයි මෙතනමයි ඉන්නේ. ඒක හොඳට තේරුම් ගන්න. You have to measure the sugar volume or the reducing sugar or the glucose that is required to reduce Cu2+ in the 5 milliliters of Fehlings A. I'll explain what is the reason for adding pumice stones. I will explain reason for each and every step. Okay. මෙතනදී වෙන්නේ සල්ලෝ තේරුම් ගන්න. ඔයගොල්ලෝ දන්නවා Fehlings A වල තියෙන්නේ මොනවද? Fehlings A වල තියෙන්නේ copper sulfate. ඒ කියලා කියන්නේ Cu2+. Fehlings B වල තියෙන්නේ sodium potassium tartarate. Sodium hydroxide. හරිද? Fehlings A වල තියෙන Cu2+ අපි මෙතනදී මේ එක්ස්පෙරිමන්ට් එකේදී Fehlings A හරියටම මිලිලීටර් 5ක් මැන ගන්නවා. එතකොට මේ මිලිලීටර් 5 තියෙනවා යම් කිසි ප්‍රමාණයක් Cu2+. එතකොට ඒ Cu2+ Cu+ නැත්නම් සම්පූර්ණයෙන්ම reduce කරන්න අවශ්‍ය වෙන sugar solution නැත්නම් reducing sugar නැත්නම් glucose ප්‍රමාණය තමයි අපි මෙතන මයින්නේ. Cu2+ Cu+ කරන්න සම්පූර්ණයෙන්ම convert කරන්න අවශ්‍ය වෙන glucose ප්‍රමාණය තමයි මෙතන මයින්නේ. අපිට ඒකෙන් තමයි calculation එක කරන්න පුළුවන්. හරිද? ඒ කියන්නේ අපි මෙතන conversion factor එක විදිහට පාවිච්චි කරන්නේ මිලිලීටර් 5ක තියෙන Fehlings ප්‍රමාණය නැත්නම් මේ 5 milliliters Fehlings A solution එක සමානයි කියලා අපි හිතනවා 50 milligram of glucose. ඒ කියන්නේ මිලිලීටර් 5ක තියෙන Fehlings නැත්නම් Cu2+ reduce කරන්න අවශ්‍ය වන glucose ප්‍රමාණය මිලිග්‍රෑම් 50ක්. අපිට ඒකෙන් තමයි calculation එක කරන්න පුළුවන්. Okay, don't ask me why we use the conversion factor. This is found by doing a series of experiments and come up with this conversion factor. Okay, this has not done by me, but the founders of this biochemical experiment. Okay.
Excuse me, sir.
Yes.
Sir, indicator එකට based වෙන්නේ නැද්ද මේ ready මේ copper 2+?
Indicator එකට
based වෙන්නේ නැද්ද copper 2+ මේ reduce වෙන්නේ නැද්ද indicator එක නිසා?
indicator එක methylene blue තියෙන්නේ oxidized state එකේ කොහොමද? එතකොට දැන් අපි දන්නවා redox reaction එකක් වෙන්න oxidizing agent එකක් තියෙනවා reducing agent එකක් තියෙනවා. එතකොට methylene blue Fehlings A එතන reaction එකක් නැහැ. මොකද දෙකටම වෙන්න පුළුවන් reduce වෙන එක. පැහැදිලිද? දෙකටම වෙන්න පුළුවන් reduce වෙන එක. නමුත් අපි කියන oxidizing power එක වැඩි Cu+ වල Cu2+ වල methylene blue වලට වඩා. එතකොට reducing sugar එකත් එක්ක මුලින්ම ප්‍රතික්‍රියා කරන්නේ Cu2+ නැත්නම් Fehlings A. ඒ Fehlings A ඉවරනට පස්සේ තමයි methylene blue එකත් එක්ක react කරන්නේ. පැහැදිලිද? මෙතන අපි අර වෙන ඉන්ඩිකේටර් පාවිච්චි කරනවට වඩා වෙනස්. ඔයගොල්ලෝ දන්නවා පිනොප්තලීන් වල කලර් චේන්ජ් එකක් දෙන්නේ pH value එක වෙනස් වුණාට පස්සේ. නමුත් මෙතන වෙන්නේ ඇත්තටම redox reaction. Okay, I will explain the redox reaction here. Then you will understand that properly. මේක තමයි main redox reaction එක. glucose තියෙනවා. glucose කියලා කියන්නේ reducing sugar එකක්. reducing sugar කියලා කියන්නේ ඒකට අනිත්‍යව reduce කරන්න පුළුවන් මේක oxidize වෙනවා. මෙතන බලන්න CHO නැත්නම් aldehyde එක COOH carboxylic group එකක් විදිහට oxidize වෙලා තියෙනවා. Cu2+ Cu+ බවට පත් වෙලා තියෙනවා. ඒක bitrate color precipitate එකක් විදිහට අවක්ෂේප වෙනවා. අපි heat එක provide කරනවා. Okay, then this is the main redox reaction. මේ Cu2+ තියෙන්නේ Fehlings A වල. අපි Fehlings A දාන්නේ හරියටම මිලිලීටර් 5යි. එතකොට අපි මේ titration එකේදී මයින්නේ හරියටම Fehling's A මිලිලීටර් 5 reduce කරන්න අවශ්‍ය වෙන glucose volume එක. මොකද අපි බියුරේට් එකේ තියෙනවානේ අපිට හරියටම දාන glucose ප්‍රමාණය මයින්න පුළුවන් නේ බියුරේට් එකේ රීඩින් එකේ. අපි ඒක තමයි මේ titration එකේදී මනින්නේ. හරිද? එතකොට endpoint එකේදී මේ you know this already. Okay, you have learned about this already. And what is happening in the endpoint is that methylene blue, which is in the blue color, will reduce into leucomethylene blue, that is colorless. Okay, that is a kind of reduction. එතකොට reducing agent නැත්නම් oxidize වෙන්නේ reducing sugar එකම තමයි. CO2+ ඔක්කොම ඉවර වුණාට පස්සේ එතන reduce වෙන්න දෙයක් නැති හින්දා methylene blue leucomethylene blue බවට පත් වෙනවා. පැහැදිලිද? Then this leucomethylene blue is very unstable. Okay, with reacting the atmospheric oxygen that can convert into methylene blue. Therefore, we have to provide a heated condition. අයි අපි මේක රත් කරන ගමන් titration කරන්න හේතුව ඒක. Okay, there are other reasons also. I will tell you that also. The primary reason for using heated conditions in this titration is that methylene blue, the leucomethylene blue, that is the endpoint that is used to identify the endpoint, can easily react with atmospheric oxygen and oxidize into methylene blue again. Okay, if you when you are doing this experiment, if you keep that after doing the titration, if you keep that aside, then definitely you will observe again blue color. පැහැදිලිද? Fehling's AB mix කරලා තමයි දාන්නේ. අපි දන්නවා Fehling's B වල ඇත්තටම ඔයගලන්ට එදත් කිව්වේ Fehling's B වල ඔයගලන්ට හිතන්න බෑ ලොකු ප්‍රයෝජනයක් නැහැ කියලා. මොකද මේ redox reaction එකේදී කිසිම සහභාගීත්වයක් නැහැ Fehling's B වල. ඇත්තටම Fehling's B තියෙන්නේ ඇයි? මම මේක explain කරා ඔයගලන්ට කලින්. Fehling's B තියෙන්නේ cupric oxide form වෙන එක වළක්වන. Fehling's B නැත්නම් potassium sodium tartrate මේකත් CO2+ complex එකක් හදන නිසා cupric oxide form වෙන එක වළක්වනවා. cupric oxide කියලා කියන්නේ මේ කළු පාට අවක්ෂේපය. එතකොට මේකේ අපිට කිසිම දෙයක් බලා ගන්න බෑ. Okay, then we add the importance of having potassium sodium tartrate is that it forms a complex, and therefore it prevent the formation of cupric oxide. Okay, so that is for Fehling's, that is the use of Fehling's B. Okay, actually that's why I told you in this experiment you have to very accurately measure Fehling's A. Okay, for that it's better to use a bulb pipette, but Fehling's B, it's okay to use measuring rate. Okay, but the accuracy should be there in measuring Fehling's A, because we assume that 5 milliliters of Fehling's A is equal to 50 milligram glucose. That is the base for our calculation. Okay, and yes, then this is the endpoint. Methylene blue will convert into leucomethylene blue, and as our demonstrator explained, at the endpoint you have to observe the bubbles. You have to observe the color of bubbles. At the endpoint you will observe very clearly color of the bubbles will turn into colorless. Okay, before that color of that bubbles are blue because of having this methylene blue. Okay, අපි දැන් මෙතන මම කලින් කිව්වා වගේ කලිනුත් කිව්වා මේ ප්‍රැක්ටිකල් එක කියද්දි අපි මෙතන titrations කිහිපයක් කරනවා. පළවෙනි titration එක අපි සාමාන්‍යයෙන් ගණන් ගන්න නැහැ. ඒක අපි කරන්නේ approximate endpoint එකක් හොයාගන්න. පැහැදිලිද? අපි පළවෙනි titration එකේදී මුලින්ම methylene blue indicator එක දාලා කරනවා. ඊට පස්සේ දෙවෙනි titration එකේ ඉඳන් methylene blue indicator එක දාන්නේ ඔයගොල්ලෝ හරියට වීඩියෝ එක බලුවා නම් endpoint එකට මිලිලීටර් 3-4ක් තියෙනවා. okay endpoint එකට මිලිලීටර් 3-4 මොකද අපි endpoint කියන්නේ approximate endpoint එක දැනගත්තා. ඊට පස්සේ අපිට titration එක බය නැතුව කරන්න පුළුවන්. හිතන්න අපි approximate endpoint එක 10 මිලිලීටර් කියලා. එතකොට අපිට 7 මිලිලීටර් විතර වෙනකම් කෙලින්ම sugar add කරලා ඊට පස්සේ indicator එක දාලා හිමින් සැරේ sugar add කරලා මේ sugar add කරන ගමන් color change එක බලන්න පුළුවන්. එක හේතුවක් තමයි even though we assume that even though the oxidizing power of Cu2 plus is higher than that of methylene blue, even small amount of methylene blue can react with the reducing sugar. Okay, that amount is very negligible, but in order to increase the accuracy of this experiment, we add methylene blue 2 or 3 milliliters before the endpoint. Okay, we do the first titration to cater that need. Okay, one reason is that. And okay, and also we have to provide heated conditions in this titration. One thing is to prevent the formation of methylene blue, again form methylene blue by reacting oxygen. And you know that when we provide in heated conditions, we can remove the dissolved oxygen, as well as that helps to increase the rate of reaction of the redox reaction, as well as it helps to release the bound sugar that is in the sugar solution. You know, food is a complex material. If you use a food material, so when the providing heated conditions will help to release the bound sugar. Okay, that is another reason for using heated conditions. Okay, and when you're doing this titration, so I'll go back to our procedure. I'll give you enough time to ask any questions, and please understand this clearly. Okay, when you're doing this titration, you have to think about several things. First thing is, you know that we use heated conditions. We do this titration during the heating of this conical flask. Okay, then you have to do this within a short period of time. Otherwise, the volume we take in the burette will not be accurate, because you know that when we provide heat, these glasswares can be expanded. Okay, especially the burette. Okay, these are the volumes of burette are designed to correct at a room temperature of about 25 degrees Celsius. So when we provide higher temperature, you know that when we doing this in heated conditions, the burette can be expanded, thereby the readings may be incorrect. Therefore, you have to do this within short period of time, and we can achieve that by first doing a titration to get approximate endpoint. Then after we can hasten the process. And also you should not do the titration continuously for a longer time. Okay, if you feel that the endpoint is it may consume lots of sugar solution, so it's better to keep that aside from the heated conditions, and after cooling the burette, then again you have to continue that in order to prevent the expansion of glassware. Also, when you add in methylene blue indicator, add that directly to the solution, not along the walls of the conical flask. Okay, then it can precipitate, and we can't observe proper color change. Other than that, when you add in sugar solution to the conical flask, you have to add directly to the solution, not along the walls of the conical flask, because it can evaporate and it will never go to the solution. Okay, you should think about those things. So we use fumous stones to provide even heat distribution. Okay, we use fumous stones. It's a kind of few small pieces of stones specific stones, not these granites, and we add few fumarous stones to provide even heat distribution to the solution in the conical flask. Okay. The second reason for using a heated condition is that if we are given with a food sample, you know that food is a complex matrix. Okay, we cannot have carbohydrates, proteins, moisture, lipids separately. So it's in a complex material. So we provide heated conditions to facilitate the releasing of bound sugar. Okay, when we are using a food sample, that helps to release bound sugar. Okay, you know that reducing agent is the reducing sugar. So there should be reducing sugar to have this redox reaction. Okay, so that's the reason. Second reason. First reason, you know that to remove dissolved oxygen. If there's dissolved oxygen, so leucomethylene blue again can convert into methylene blue. Okay. Okay. So I do you have calculator? I'll give few minutes. If not, take your calculator and a piece of paper and a pen. So we will do the calculation here. Okay, I will provide you readings, and also you have to submit this as a your practical record, estimation of glucose, and also please careful and understand this, because this is very important. Okay, there are only few calculations that we can do using these biochemical experiments. I mean the things we have in the syllabus. This is one of them. Okay. Okay. Then we prepared the sugar solution to be tested by dissolving 2 grams of the given impure glucose sample in 250 milliliters of distilled water. Okay. So record this conversion factor is 5 milliliters of Fehling's A is equal to 5 milligrams of 50 milligrams of glucose. Okay. Record this sugar solution was prepared by dissolving 2 grams of the given impure glucose sample in 250 milliliters of distilled water. Okay. Okay, you can do whatever you want, but I said a few times that this is important. It's up to you to how you grab the things, but I can just advise you. Please understand this carefully and know the calculation. Okay. And you know you are adults and you can do whatever you want. And I highly can I say I highly appreciate the freedom of young youth, and you can do whatever the things, but when you are learning, pay attention and grab the things clearly. Okay. So here are the titration readings. First reading was 6.50 milliliter. Second reading was 10.55 milliliters. Third reading was 10.60 milliliters, and the fourth reading was 10.35 milliliters. Okay. So when you are doing the calculation, are you going to take all the 4 readings? So what are you going to exclude? Okay, I going to exclude. Definitely, we have to exclude the first reading. You know that there's a huge difference between the other readings and the first reading, and you can consider these 3 readings. Okay, you can consider these readings also. It's quite a little bit deviate than the first 2 and 3rd readings, but it's okay. You can use the 2nd, 3rd, and 4th readings. Okay, 10.55, 10.60, 10.35. Okay, so actually, if you want to exclude a reading, you have to do a Q test and see whether one is significantly deviate than others. And as we can see, 6.50 is significantly deviate than others, and definitely we have to exclude that. Actually, in this titration, specifically in this Lane-Eynon method, we don't use the first reading because it is done to just get an approximate idea. Okay, then you can take the other 3 readings, 2nd, 3rd, and 4th reading. Okay. Then we will move on to the calculation part. Okay, then what is the average endpoint of the titration? So I think you have a calculator with you. Please then answer me. What is the average endpoint? It's 10.5. Okay. Then you have to take the sum of these 3 the readings we're going to consider, and you have divided by 3. Okay, then you get 10.50. Then the amount of glucose in 10.5 milligrams of the sample is 50 milligrams. How do we come up with that? How do you come up with that? How can we come up with that? Amount of glucose in 10.5 milliliters of the sample is equal to 50 milligrams. You know that we used a conversion factor. 5 milliliters of Fehling's A equal to 50 milligrams. And you know that, and through this titration, we got that to reduce completely 5 milliliters of Fehling's A, it consumed 10.5 milliliters, 10.50 milliliters of the sample. Then we can say that amount of glucose in 10.5 milliliters of the sample, it means the sugar solution, is 50 milligrams. Okay, so that's the use of conversion factor here. You have to understand that clearly. It's using the conversion factor. Good. Then amount of glucose in the sample. So we in the first I mentioned you that to prepare the sample, we dissolve to 2.0000 gram of the given sample in 250 milliliters. To calculate the amount of glucose in the sample, we have to multiply it by 250 milliliters, because from that 250 milliliter you got some amount to the burette. Okay, so we calculated that among the 10.5 milliliters of the sample, it contained 50 milligrams of glucose. Then we can calculate the amount of glucose in the total sample. Then what is the answer for this? This is a simple calculation, but you have to understand this clearly. 1190.47. Is it from what is the unit? Is it milligrams or grams? It's milligrams. 1190.47. Okay, good. Then it's in milligrams. Yes. If it convert that into grams, it's about 1.1905 grams. Then you can calculate the percentage of glucose in the given sample. So how do you calculate that? You know that we took 2 grams of the sample and dissolved in 250 milliliters, and we calculate the glucose amount in this 250 milliliters. Then you can express that as a weight to weight percentage. So glucose percentage of the given sample is that. What is the answer for that? Yes, most of you have come up with the correct answer. So I think this needed to be corrected. What is the answer? Yes, 59.53. 59.53. Okay, then if you consider about significant figures, you have to use 5 significant figures here. Okay. Okay. Then so that this is how we do the calculation. Then other than that, do you have any questions? Please understand this clearly. You will need this definitely. And also, this is not a very accurate method, but to have a approximate or rough idea about what the glucose content in the given sample, we, as a simple biochemical test, we can use this biochemistry experiment. We can use this. Okay. So what are the drawbacks of this test? You can think about that also. What will be the disadvantages of this test? Can you think? In here, we think that only reducing sugar in the sample is glucose, but it can't be. So there are other types of reducing sugars. You know that all monosaccharides, disaccharides, some disaccharides can act as reducing agents. So in this experiment, when we estimation the glucose concentration, we think that in the sample only reducing sugar is glucose. Okay, but we do this test to for samples that are high in glucose, but there can be other types of reducing sugars also. So that's a kind of drawback in this test. Okay, other thing is that even though we assume that methylene blue react at the end, it can react with the reducing sugar or the glucose while doing that titration, but the amount is negligible. Okay, it will not affect to the accuracy of this test significantly, but some amount can be reacted. Okay, what are the other things?
රත් කිරීමේදී වාෂ්ප වීම
Oh, රත් කිරීමේදී වාෂ්ප වෙන්න පුළුවන්. No. එහෙම හිතන්නත් පුළුවන්. එකක් තමයි අනිත් එක තමයි මේ පත් කරනකොට glass vessel වල volumes expand වෙන්න පුළුවන්. එතකොට අපි දැන් burette එකේ ගන්න reading එක හරිම reading එක නොවෙන්න පුළුවන්. මොකද ඒක expand ලා තියෙන හින්දා volume එක වෙනස් වෙන්න පුළුවන්. ඒකින් හින්දා අපි පුළුවන් තරම් expand කරන්න ඕනේ. ඒක බලපං. Because we can do some proactive things to avoid the errors that can occur in this experiment. One thing is adding methylene blue just before the endpoint and doing the titration within a short period of time without exposing the burette to a high heat. Okay, and to provide even heat distribution, we use pneumocystons. Okay, did you understand this properly? So make an endpoint එක හයට තේරුම් ගන්න. endpoint එක වෙන්නේ dictate precipitate එක හැදෙන තැන නෙමෙයි. ඒක ගොඩක් කට්ටිය වරද්ද ගන්න තැන. dictate precipitate එක හැදෙන තැන නෙමෙයි endpoint එක වෙන්නේ. dictate precipitate එක ගොඩක් කලින් හැදෙන්න පුළුවන්. හරිද? dictate precipitate එක කියලා කියන්නේ Cu plus Q plus hydroxide එක රත් කරනකොට Cu Q plus oxide හැදෙන්න පුළුවන්. ඒක dictate කියලා කියන්නේ ඒක නෙමෙයි endpoint එක. endpoint එක වෙන්නේ අර bubbles colorless වෙන තැන. Okay, so you have to observe carefully the color of the bubbles. Okay, then why heated condition is provided in this test? One reason is that if we use a food sample to aid or to facilitate the releasing of bound sugar, sorry, not bound water, bound sugar. Okay. Other thing is that this leucomethylene blue, if dissolved oxygen is there, leucomethylene blue can readily oxidize into methylene blue again. Okay. To remove dissolved oxygen, we use heated conditions. Okay. So we use a solution, even in sugar solution, the dissolved oxygen can be there, and also in the Fehlings. Okay, to remove the dissolved oxygen and to avoid forming again methylene blue from leucomethylene blue, we use heated conditions. Those are the two major reasons for using heated conditions in this experiment. Okay, you no need to think remember about the structures, but understand the principle. That's it. Other than that,
Excuse me, sir.
yes. Oh,
Sir, when our first titration is just approximation one,
yes.
is it necessary to add methylene blue?
Yes.
I mean that the Cu2 plus reduce into Cu plus, then it's always give a color change, no?
Cu2 plus will turn into Cu plus, and how can we determine the endpoint? Endpoint should be definite. Cu2
Oh,
plus
I
Cu plus.
thought it just approximation.
Cu2 plus Cu plus වෙනවා. එතකොට reducing sugars 1 milliliter add කරත් ඒක වෙන්න පුළුවන් නේද? අපි 2 milliliter add කරත් ඒක වෙන්න පුළුවන්. අපිට end point එකක් අඳුර ගන්න බෑ. So in order to identify your endpoint, we have to have a mechanism. Usually we in this kind of experiments, in usual titrations, we use an indicator for that. Okay, so methylene blue is that kind of an indicator, but the mechanism is different than other acid-base titrations. Okay, in acid-base titrations, you know that the indicator will give a color change depending on the pH range of the solution. Okay, but in here, it's a kind of redox reaction. And other than that, there are different kinds of titration, potentiometric, electrochemical, and many types of titrations. This is also a special type of indicator use. Okay, in the first titration, we add indicator in the very initial stage, because we don't know what amount of sugar will consume, or what will be the burette reading. So we add the methylene blue at the very first stage. Okay, so we can have a endpoint. ඔගොල්ලෝ මේක හරියට ඔබ්සර්ව් කරා නම් මුලින්ම Fehlings A Fehlings B දැම්මට පස්සේ Persian blue color එකක් එනවා. ඊට පස්සේ අපි sugars add කරද්දි drops කිහිපයක් add කරාට පස්සේ briquetted precipitate එක පේන්න පුළුවන්. අර cortical flask එකේ. ඒතර අපිට endpoint එකක් හඳුනා ගන්න බෑනේ හරියටම. So do the calculation, we have to get an endpoint. Okay, so
If Cu2 plus is responsible for the Persian blue color, can't we assume that when the sugar solution totally reacted with copper ion, then the Persian blue color totally get removed and it's turned into colorless in the Fehling solution?
yes, I totally got your idea. The thing is, even though we add 1 drop of sugar, the redox reaction can happen. Redox reactions mean glucose can oxidize into gluconic acid, and Cu2 plus will reduce into Cu plus, and it will not remain as Cu plus. It will, due to the presence of hydroxyl ions, it will form Q plus hydroxide, and in the heated condition, it will convert into Cu2O, means Q plus oxide. That is the briquetted precipitate. Even when we add in 1 drop of sugar, that Cu plus can form. Okay, So we cannot identify a definite endpoint there. Okay, then the Persian blue color and the pictate precipitate. So we cannot identify it definitely what will be the endpoint. Okay, පැහැදිලිද? ඒ කියන්නේ අපිට එතනදී අඳුර ගන්න බෑ මොකක් මේ ඒක ඉවරයක් වෙන්නේ කොතනද කියලා. Persian blue color එක සම්පූර්ණයෙන්ම අයින් වෙන තැනක් අපිට හොයාගන්න බෑ. මොකද sugar solution එක පොඩ්ඩක් ඇඩ් කරා අර CU මේ redox reaction එක වෙන්න පුළුවන්. අනිත් එක prectate precipitate එක හැදෙන්න පුළුවන්. එතකොට අපිට අවසානයක් හොයාගන්න බෑ. මේ titration එක කරද්දී Persian blue color එක නැති වෙන තැනක් හොයාගන්න අමාරුයි. එතන blue color එක වෙනස් වෙනවා කියලා කියන්නේ අර Fehling solution එකේ තියෙන blue color එක නෙමෙයි methylene blue එකේ තියෙන blue color එක. Okay. Bubbles වල color එක බලන්න අපි ඒකයි. අපි solution එකේ color එක නෙමෙයි බලන්නේ bubbles වල color එක. අපි ඒකයි methylene blue just before endpoint වලදී add කරා. පැහැදිලිද? Is that
Yes,
clear
sir.
for you? Is
Yes,
that
sir, I
clear for
got
you?
it. Yes, thank you. Thank you, sir.
Okay. Yes, the specific name for this test is I will type that in the test Lane and Eynon test. Yes, Lane-Eynon test. Lane-Eynon test. අපි ඒක මම තනියක founder ගේ නම වෙන්නේ නැති සම් founder කෙනෙක් කර දෙන්නේ කරේ. අපි සාමාන්‍යයෙන් ඔය Lane-Eynon කියන නමක් පාවිච්චි කරනවා මේකට. Okay. මේක accuracy එක එච්චරම වැඩි නැහැ. නමුත් to have an approximate idea, rough idea, and using the equipments we have in the laboratory, we just use the Fehling solution and methylene blue, which is a abundant indicator, and just a burette conical glass and the basic glassware. So using that, we can have an idea about the glucose content. You know that glucose content in samples for glucose content is a very good indicator, especially in health sector. You know that it's a widely testing health indicator in blood and urine. Okay, so actually, if you need to measure glucose content very accurately, there are advanced biochemical assay kits. When we don't have such things, we can just use this test. Okay, just to have a rough idea. Okay, so in that sum, how to got the weight of glucose as 50 milligram, because the here volume is 10.5 milliliters. In that sum, how to get the weight of glucose as 50 milligram. ඒක අපි මේක ගන්නේ ඔයාලට තියෙන ප්‍රශ්නේ අපි මේ 10.5 sample එකේ 50 mg තියෙන්නේ කොහොමද කියලා ගන්න එක නම් අපි ඒක ගන්න මේ conversion factor එක. අපි conversion factor එකේ හිතන්නේ 5 milliliters of Fehling A is equivalent to 50 mg of glucose. Then this much of glucose was consumed to reduce 5 milliliters of Fehling A. Then 5 milliliters of Fehling A equal to 50 milligram of glucose. That is the conversion factor. So there we use the conversion factor. Okay, that is the conversion factor we require. That is why we the conversion factor we require to do this calculation. Okay, මෙතන හිතන්න මෙහෙම ගත්තේ මේ titration එකේ average value එක 10.50 මේ 10.50 තමයි අපිට consume වෙලා තියෙන්නේ Fehlings A වල තියෙන Cu2+ ඔක්කොම ටික Cu+ බවට පත් කරන්න. එතකොට Fehlings A 5 ml වල අපි හිතනවා 50 mg glucose තියෙනවා. ඒ කියන්නේ මේ sample එකේ 10.5 ml වල 50 milligram glucose තියෙනවා කියලා අපි හිතනවා. That is the conversion factor. Okay. එතකොට 10.5ක 50 milligram නම් තියෙන්නේ 250 කොච්චරද කියලා බලන්න. Very simple. 250 ගත්තේ 250 කියලා කියන්නේ අපි මෙන්න මම ඒකයි ලියා ගන්න කියලා කිව්වේ. ග්‍රෑම් දෙකක් 250ක දිය කළා තමයි අර sugar solution එක හදාගත්තේ. ඒකෙන් ප්‍රමාණයක් තමයි burette එකට ගත්තේ. ඩයෂන් 5ක 50ක් තියෙනවා නම් 250ක කොච්චරද? 250ක මෙච්චරක් තියෙනවා නම් ග්‍රෑම් දෙක කොච්චරද? ඒක තමයි glucose percentage එක. Okay, so other questions. That's okay. If you have any questions, just ask. Since you have no other things to clarify, I will end the session by here. And you have to submit a practical record for this. I will upload the things in the LMS. And okay. So I'll stop by here. Thank you.